In this video, we're going to talk about task-driven seeing, and we're going to use the example of disentangling wires. This example is from a paper 3 years ago, which really opened my eyes to the way that the primary visual cortex works and how it's used as a multi-scale cognitive blackboard. I find this example of task-driven seeing particularly interesting. So the task is set up as follows. The monkey or person has to look at two devices with slightly tangled wires. On the left we have the toaster and the hairdryer. On the right we have the plugs that plug into the wall, and we have to figure out which wire leads to which device so that we plug in the right device. We've got some labeling here that we'll need to understand in order to appreciate the following slides. So on the left we have two Roman numbers 1 and 2 as labels. 1 represents the wire coming from the toaster, and later on, as we trace through the wire from left to right, from the device to the wall plug, 3 represents the same wire. Conversely, 2 and 4 represent the wire for the hairdryer, and 4 is going to be later in the process because we're going to trace the wire through from the device to the wall socket. So here's a slide, and it's a little bit complicated, so I'm going to walk through it with you. We're talking now about neurons in area V1, and what we see here is that for both neurons that are early in the tracing process, i.e. areas 1 and 2, close to the devices, and neurons that are late in the process, areas 3 and 4, close to the wall plug, they both have a high activation at the beginning, which is very fast, less than 100 milliseconds, and this basically represents the information of the stimulus from the eyes. So this activation represents a new image that is falling on the portion of the retinal field that's relevant to these neurons. So we have this high activation, and then the lines that follow, the yellow and black lines follow. Looking at the left panel, for instance, we see that the target, which in this case is labeled 2, the yellow line, is more activated, and for the distractor, the activation drops off faster. So that gray region between the yellow line and the black line here represents the extra processing and the extra attention devoted to the wire that they have seen as the target based on the task provided to them. Okay, so what we see here is the fact that the activation follows the structure of the task. So on the left, we have more activation starting early because the region we're looking at for labels 1 and 2 is to the left of the figure, and that's where they're starting from. Whereas on the right, we see the activation as difference as reflected in the gray area between the target line, yellow line, and the distractor, black line. We don't see this distinction occurring until after 370 milliseconds, showing us that it takes them about 270 milliseconds to scan from the left side of the wire, represented by labels 1 and 2, to the right side of the wire, represented by labels 3 and 4. Okay, to summarize then, we are seeing a difference in activation between the target and distractor wires, and the timing at which this difference in activation starts depends on how long it takes to reach the particular neuron as the person or monkey traces the wire from left to right. In this figure, we're looking at the size of retinal field at the different layers. So we have at the bottom of the figure area V1, And this, because it's the primary visual reception area, it's going to have small receptive fields because there's been very little visual processing thus far, and it has the highest resolution in the visual processing system. Area V2 is the next stop, and because it is now abstracting some of the information, it's going to have larger receptive fields in the retina. And what this means is that it is covering, each neuron is covering a larger part of the retina because it's receiving inputs from a larger portion of the retina. So this is kind of a hierarchical system, so that the inputs from V1 go to V2, but multiple receptive fields in V1 will report to the same receptive or retinal fields in V2, and thus the receptive fields in V2 get bigger. And this process occurs again as we transition from V2 to V4, so that by the time we get to area V4, the receptive fields are quite large. Now what this means is that if the wires are not too badly tangled at a particular point, and there's a large separation, we can go through and do the processing at area V4. So in this case, we see that there are two yellow circles on the left of V4, and we can, because the wires do not overlap on these receptive fields, we can process at that level and process quickly. However, by the time we get to the middle region, we see that the receptive field is now red at area V4, meaning that the two wires overlap, and the system can't tell them apart because they're both activating the same neuron with the same receptive field. So then we have to go down and go to a lower level, and ultimately we go down all the way to V1 in order to do the processing, because here the visual fields or the receptive fields are small enough that the wires are not overlapping and they can be differentiated. So over time, you'll see that the if we go to the right side of the figure, again, when we get to the end, the receptive fields are now large enough that area V4 can handle the disentangling and do it faster. So there is the process of disentangling moves up to a higher level. So to summarize, when we are trying to do a fine visual task, we have to go to the right area in the visual processing hierarchy. When the task can be done with large receptive fields, in other words, that we don't need a particularly fine distinction, we can go to a higher processing level and the task can be done faster. On the other hand, if the task requires a very narrow or small visual distinction to be made, then we need small receptive fields or retinal fields. And this puts us back at area V1, and because we're going to have to go through many different neurons as we trace out the difference, the task is going to take longer. In this final slide, we're going to be looking at eye movements. And the idea here is that if the curves are far apart, the spread of object-based detection has similar timing in V1 and in an area called the frontal eye fields. So the frontal eye fields are used for planning eye movements. Now, eye movements are actually controlled in the brainstem. So there's a region in the brainstem that actually guides the eyes and tells them where to go. But the planning, the task-based planning for that those movements occurs in humans and monkeys in the frontal eye fields, which is part of the prefrontal cortex. So it seemed like with earlier and more primitive organisms that don't have this kind of field in the prefrontal cortex, there would be more sort of instinctive eye movements based on very simple principles. But if the animal is actually doing more planning, then we need the executive control in the prefrontal cortex to help impose the task on the visual scanning. So here we see that both the frontal eye fields and the area V1 are being activated at around about the same time, around about 100 milliseconds. And we also see that the distinction between target and distractor in terms of activation occurs at roughly the same time of 160 milliseconds. So finally you'll notice that the distinction between target and distractor is greater. The gray area is greater in the frontal eye fields, showing the task-oriented nature of the frontal eye field. So they're really focusing on the difference between the target and the distractor, and really emphasizing the position of where the target is and giving it much greater activation. In summary then, task-driven seeing is a process where we are using the cognitive blackboard and we're using other parts of the brain like the frontal eye fields to create scanning strategies that will spread visual attention or object attention over the portions of the visual field where the task requires us to look and where we have to make visual decisions associated with the task. The disentangling wires task is a good one because it allows us to see the effect of what happens when there is more or less visual distinction between the wires that have to be disentangled, and we can see how the organism, in this case, an ape or a monkey or a person, deals with different amounts of entanglement.